Hello everyone, we are about to start this webinar shortly. We are waiting for people to join. Hello everyone, so people are still joining us. Maybe we can wait for 5 more minutes. Greetings everyone. I am Dr. Aditi Nag. I am Assistant Professor at Dr. Bilan Institute of Biotechnology, Jaipur, and will have the honor of moderating this webinar today. So as we all know, we are gathered here to find out about an innovative approach which can revolutionize our fight against COVID-19 pandemic in India, as this is able to detect the upcoming COVID-19 wave as early as 20 days before the actual rise in the number of cases, of patients, and thus it can give us a timely alert. Joining us from this informative session today are Professor Paramita Chakrabarty, who is an associate professor at SRM Research Institute and Department of Civil Engineering in SRM Institute of Science and Technology. As a leader of environmental science and technology research group, she relentlessly pursues high quality research on fate and transport of legacy and emerging contaminants under varied climate regimes in India and other parts of Asia. Her group is also working on cost-effective remediation and plastic waste recycling techniques for the betterment of society. We also have with us today Dr. Wang Jiang, who is a senior lecturer, School of Civil, Mining and Environmental Engineering at Wollongong University, Australia. He has broad research interests in wastewater treatment, infrastructure management, environmental health, microbial corrosion, and smart water. And he has been working with the WB approach since even before the pandemic has hit the world. I would also like to take this opportunity to introduce Dr. A.B. Gupta, sir. He is a professor of HAC Department of Civil Engineering, Malviya National Institute of Technology, India. He is working on various aspects of water and air pollution and their impact on human health. Also evolved pathophysiologies of fluoride, nitrate, and aluminum toxicities and linkage between air pollution and human respiratory health. He has developed low-cost technologies for removal of fluoride and nitrates from drinking water and wastewater. He has also done advanced bioprocesses for waste treatment, characterization of particulate matter fractions in air, developing field kits for biological analysis, and many areas of research addition to all these. And last but not in the very least, our own Dr. Sudipti Arora. She is a research scientist here and assistant director at Dr. Bilal Institute of Biotechnology, India, who is the pioneer of wastewater-based epidemiology study of SARS-CoV-2 in this capital city of Rajasthan, that is Jaipur. She is also involved in applications of institute and village level based zero waste model by providing sustainable technologies like permafiltration, bioremediation, constructed wetlands, and organic farming. I would like to welcome everyone on behalf of Dr. Bilal Institute of Biotechnology for this international webinar on wastewater-based epidemiology prediction. So moving on, the challenge of COVID-19 detection is that not only symptomatic patients, but also asymptomatic and mildly symptomatic patients are also present and affected with this disease. And although these are asymptomatic and cannot be detected usually, these individuals can still quickly spread the disease before they are even tested. So it is a big threat, and that's why it has led to this globally blown pandemic. The idea behind WB to attack this disease is that any pathogen, be it SARS-CoV-2 or any other pathogen which affects an individual, whether symptomatically or asymptomatically, will be shed through human excretions, and if these excretions are dumped into the wastewater, they get collected over there into the wastewater on a community level. Now, if such a pathogen is present or prevalent in any human or individuals or in a community, it can be detected by a sensitive detection tool through even a single test. So therefore, there is no need to do individual level testing, and just a single test can give us a prediction or an indication as to where the next hotspot can be. Okay, so now speaking of advantages, what if I could tell you that you can not only know where the next hotspot community for pandemic is in a cost-effective manner, but you can also find out how much coffee our neighbors are drinking during this stress. Now, this magic of WB approach is there, and to tell us more about it, I would like to invite our first esteemed speaker, Dr. Chakrabarty, who is the principal investigator for several national and international projects, including ongoing projects with premier institutes of USA, Norway, Switzerland, and Japan. I would like her to speak on the knowns and unknowns of the pandemic, a cost-effective technique for early signals. Please, ma'am.
Thank you, Dr. Aditi Nag, for this warm welcome. And thanks a lot, Dr. Siddhi Arora, for inviting me in this forum. It's like my privilege to be here today. Please allow me to share my screen. I hope my screen is visible and I'm audible.
Yes, ma'am.
Good morning to all of you. Yes, we are in a very difficult condition, but this is a very, a seminar, a webinar, which is very important. And I really appreciate the idea of hosting such a webinar, which might be important for the next forthcoming webs. So with my title, it is understood like something that we know and something we don't know. I will, in the initial part of my talk, I'll just discuss something that we know and a little bit what may not know, may not all of us know about it. And then what like SIM-WB technique, how we have used in Chennai. When it comes to the epidemics, there are a lot of issues that talks about like basically we are impacting the ecosystem. When there is a damage in the ecosystem, mostly we complain that a lot of microorganisms may come out and they can impact in a different way. Although many of those organisms might be very helpful and they play a vital role for the normal functioning of all the earthly activities. So when we are hit by this pandemic, we have to also recall how we had been torturing the ecosystems in several other ways, which we are also working in some other studies. We all know that COVID-19 is the disease and which is causing the severe acute respiratory syndrome caused by this virus SARS-CoV-2. There were earlier coronaviruses also, but this one has really brought a very disastrous impact on us. The important part of the structure of the virus could be the virus spike S protein that mediates its membrane fusion by binding to the cellular receptors, which is how it functions. Could be again another important factor that we all know about. But then the important part which we need to understand is like how long this virus would survive, and which is important to know in a way so that we understand how do we handle the viral spread in different media. So there are different experimental procedures like where people have tried to understand the residence time of the virus. Like for example, 2 days in hospital wastewater. This was earlier a corona family which was seen, but it has also been seen like in respiratory specimen, it can stay for 17 days at room temperature. It's a 2020 publication. Now coming to the fact that we all know, yes, through the droplets, at a certain distance we are maintaining within the distance, if somebody is coming in between, might be getting infected, and also through the, transfer through those droplets and different surfaces that are actually impacted with the viral load. In that case, it's an interesting study I would like to share that where different types of surfaces were examined and seen how long in each of the surfaces the virus can stay. Like the residence time of the virus till the time it is able to spread. Interestingly, plastic was the major one that on which the virus can stay for longer, and stainless steel also followed by plastic, like more than nearly 5 to 7 hours. On these two matrices, the virus can stay longer. So which means like, we are using a lot of plastic gadgets all the time, including our cell phones and everything. So that on the surface, if the surface we are coming in front of the contaminated person, so it is important that we take care of those kind of plastic surfaces. This is something like not every time is to us, the residence time of the viral particles. And definitely, as Dr. Nag introduced, wastewater, it's a very, very important and very significant technique to understand the viral load. Basically also because we can get in wastewater, it's like all the 2020 papers, people have come up with a lot of publications talking about the survival of the virus. But good news is like there is no so far any intimation that there can be spread of the virus through water or wastewater. So that's good news, and which we can access and go ahead with more experiments. And definitely if pH can play an important role in the media, especially when we are going with the wastewater treatment plant, the pH can play an important role, whether the pathogen would survive or not. Other thing is also maybe if we maintain certain pH in the final part of the treatment plant, we can also take care of the removal of the virus. And ultraviolet radiation is important in a way that it can help to understand that how to what extent we can actually take care of the viral load in other surfaces. Many a times we are asked to keep the, when we are doing the experiments, we keep all the stuffs like after the post experiment in the UV radiations to disinfect it. So those kind of informations are also important, and it should be known to us while we are doing the experiment to have a successful experiment. So the need was like, especially for environmental researchers like us, that how environmental factors can be important in the transmission of SARS-CoV-2 and help the impact of COVID-19. So in this need of the art, the first paper that came, that's Warish Ahmed with the Jochen Muller's group in Australia, like it is like one of the pioneer paper, I guess, like the first of its kind that came up, extensively good work, and that gave us an idea for all of us. And so in a similar time frame, when this paper was out, like we were also seeing Metro Water in Chennai city, first time from India, like they picked a few wastewater samples, and those were found positive, and those were reported that came in the Hindu, even TOI covered it. So all this during this time, I would say frankly, like I don't have a microbiology background, but as an environmentalist, I was looking into the factor that how do I contribute? This is the high time we need to come up with something, and definitely wastewater-based epidemiology is the target. But in addition to that, I was also looking into the factor whether there is any chemical marker we can tag with the viral particles. So that was a sort of a new idea that I had in mind. Unfortunately, we were supported, like our project was, and we were supported by Swiss Agency for Development and Cooperation. SDC funded the project, and in my group, we had Mu Gamma consultants and the Energy Research Institute, Theory from New Delhi. So they were basically, Mu Gamma and Theory were taking care of the policy part, while I was taking care of the technical part of the project. So how did we go? Like, so in this discussion, next half, I'll be talking about how did we do the sampling, how did we handle the samples, and what are the results we got. Basically, in wastewater, what we are trying to track is the N1 and N2 genes that is present in the nucleocapsid gene of the virus. And we are using like any human sample. How do we do that? Similar way, we are using reverse transcriptase RNA polymerase chain reaction. And then we are trying to identify whether the sample is positive or negative. But in addition to that, we were also looking into the quantity of viral load. I'll tell you how we did it. Now, before we get into this story, we have to say, like, we were going through a lot of papers, including one important paper from the host institute, which came from your institute, and IIT Gandhinagar from India. Then also there were papers apart from Australia, the Dutch scientists who were coming up. There were papers from Paris. So different organizations were working on WB. So it was like a global initiative, I would say. So what was our main basic question? The first thing that was, as I told you, like I deal with mostly organic tracers. So it was in my mind, can we find out an organic tracer which could correlate the viral load? And then post-lockdown, whether the viral loading would change. And because we have two stagnant rivers, like Delhi has Yamuna, we have Adir and Kuam, which are extremely contaminated and mostly stagnant. So I was also having this interest that whether the community dwellers, especially where the socio-economic condition is poor, whether there is loading on the riverine load, and how does this go during the lockdown or post-lockdown phases. So these were the questions we had in mind in this project. Now, with the marker thing, like recently, I mean, this is of course published this year, but a couple of years back, I tried to understand like what could be an important marker for human waste. And caffeine we find is very, very interesting. We also try to expose like those, the compounds that we got in human cell lines. Caffeine we could very well determine as a very good human waste marker. So now I took this chance, like this experience combined with the WP. So our objectives were to measure this viral load in riverine environment, because initially we had to go through a lot of permission procedures. So we started with the riverine environment first, and then tried to work out with the sewage treatment plants and the sewage pumping stations in Chennai city, wherein Chennai Metro Water was a big support. They not only allowed us to collect the samples, but the engineers also helped us in the process, and they also co-authored with us the paper. And then apart from that, we were trying to investigate whether this organic traces, we tried with caffeine and carbamazepine. And in addition to that, we also analyzed a lot of pharmaceutical compounds, trying to understand what is the situation with those compounds during this time. So how did we collect the samples? Basically, we collected from 4 sewage treatment plants that are part of in the Chennai city. I missed showing this map. Yeah. This is like, you see, there are 4 important treatment plants in the city. We catered to all those treatment plants. And then we had SRM hospital wastewater. And nearby each treatment plant, we had sewage pumping stations. The northern side had 2 sewage pumping stations, and each STP has 1 sewage pumping stations. And we also collected water samples from these 2 rivers, Kuam and Adir. And you can see in the pictures that you see the river conditions, nowhere less than a sewer outlet. So these were the canals of the river conditions. We were collecting those samples last year. And in the sewage treatment plant, what we did is we collected samples from the inlet, then from the primary sludge and the outlet. There's also another intention was also to check, caffeine was to understand the efficacy of the treatment plants. Now, when extracting RNA, we actually tried 4 different methods. Of course, supernatant method is very well published. Many of us are well aware of this method, but then we added 3 more methods, and I will be discussing those methods in the next slides. And we wanted to go for N1, N2. In addition to that, for having a control given, we had RP as well with each of the samples. So in the supernatant method, it is like it takes 18 to 22 hours, and it involves a lot of steps. One thing I should mention that during this process, every day, early morning, we were collecting the samples, and we were trying to process the sample the same day. So we were divided in groups. We are prepared. The lab was prepared for that. And this here, basically, in this process, we will use the supernatant. And the important step is this one, where we use the American ultracentrifugal filter before we go for the RNA extraction. So each step, this filtration step, first of all, initially after adding the buffer, a filtration step, then it comes with the final stage of American ultracentrifugal filter. So this processes are very well defined in Ahmed's paper, which we followed in the first go. But then again, we went for 3 more methods. These 3 methods takes between 1 to 2 hours time. First one is the composite method. The composite method, we just take 1 ml of sample, and we then add 10 microliter of protease K, and then place it on the dry bath and subject it for RNA extraction. The sediment method, I want you to notice this because it's very important, and it is a little bit cost effective because American filter, it's pretty expensive when we are doing a lot of samples. In sediment method, we can just take a loop of the sample, again, a subject with the protease K and followed by RNA extraction. Another one, which we try to use is the syringe filter, wherein we just use syringe filter to filter 10 ml of samples was subjected to syringe filtration, and then the filtered sample was subjected to RNA extraction. Now, apart from RNA extraction, we have to mention that we followed with all the WHO recommended RNA extraction kit we were used, and we followed all the proper protocols. After the RT-PCR analysis gives us a positive or negative sample, but then we also used our nano drop to understand the viral load. So what is the quanta of the load in those samples? So what did we see? You see, like most of us, we are all aware of CT values, like cycle threshold values now. And because with human samples also, it's the same. And usually we tail it between 23 to 40 CT values are the good ones, and lower the ones are more viral load, and higher towards 40 are having lesser viral load. That's what we have the prediction. If you see here, for all the 4 methods, all the influent samples were found to be positive, but effluent samples were all negative by all the methods. This was one very important thing that we got, and which says that our STPs in Chennai city were functioning very, very ideally during those periods. So nothing was coming in the waste, in the treated water. Now, when we are trying to compare the 4 methods, this is one interesting finding we found. Like the sediment method, supernatant was good pretty much for all the samples, but the sediment method was actually much more applicable. And you see, when you don't have the Amican filtration process, so you save a lot of money in that side. So I would highlight, if there is a budget constraint, we can always go for this. And what reasoning could be given is, given the fact that SARS-CoV being hydrophobic, this possibility is there. So it can more partition onto solid matrix, which is having both the switch treatment plants and also the pumping stations. The quantity of dissolved solid or total solids are much higher in those sample matrix. But unlikely when we saw it in the hospital wastewater, which didn't have too much of total dissolved solids or solids in the matrix. In those cases, we saw like mostly supernatant samples gave the good results. And in addition to that, I would say, like the hospital wastewater, many samples had CT values less than 30. So which was again coinciding with the dates where we were having more number of patients before lockdown. So the hospital wastewater was within our jurisdiction. So we could do during the lockdown and post-lockdown. So during lockdown, a lot of patients were there in our hospital, which was actually matching with the more load in those days. And we did this sample collection on a different time interval. This was also again interesting that we found usually in the evening time there were more viral, and that was again another important point we found in this observation. And apart from the STPs and hospital wastewater, we have also checked the river samples. These are the sites. The river samples also, like hospital wastewater, we did for during COVID and post-COVID phases. So during COVID, the hospital wastewater loading was much less. If you see the yellow colors, but the brown ones are the post-lockdown. That means now people were allowed to come out. And it's unfortunate that many of the sites, we ourselves visualize open defecation. So that is also could be one reason for added load. And this area, this is the northern part, like it's like completely a slum area. We are seeing more of the viral load. Not only this, we are also seeing more of the loading of the caffeine in those areas. So this is also coinciding. Wherever we are seeing higher caffeine load, it is also matching with the viral load. What is happening with the STPs? As I said, in the STPs, in the inlet and the outlet, there was no caffeine concentration, very less caffeine concentrations. And this more than 85% removal of caffeine, it says like the viral load was also not there. Caffeine was also removed. So this was actually a kind of confirmation that the STPs were functioning very properly. We also did a lot of fluoroquinolones analyzed during this process. The fluoroquinolones were also seen pretty high, but apart from fluoroquinolones, the azithromycin as another antibiotic was pretty high because maybe the use of azithrol, which was recommended mostly for the patients, this could be a reason for that. How do we understand? Like if the marker is there and the concentration of the viral load is there, what we tried to do is a very small statistical understanding, which we use the dynamic range. Dynamic range is nothing but the ratio of the arithmetic mean of the concentration of each of these organic traces and with the RNA copies. And if you see this figure, what we are finding is like that for KFIN, with all the methods, we were seeing more than 90% coincidence. And another part is like the dynamic range is so high for KFIN, it is above 8,500 compared to all other compounds we had, which means even in diluted conditions, even if the water is diluted, still KFIN could be used as a marker to understand whether there is more human loading or not. And as we know, we all predicted the number of infections during that time using the 4 methods. And importantly, sediment and syringe method, which was much cheaper, we found that they gave very results, very good results, very close to the real number of cases we got during those phases. This is like, apart from those results, I just wanted to highlight, like we did one just remote sensing work in the Indus Ganga Brahmaputra plain, just giving a case study, like where you see that all the treatment plants are located here in the map on my right hand side. And here we tried to get through March to August, like we are trying to get the how the it is spread of the disease. This is the real time data. So if you see like where STP locations are very close nearby. So if we have a catchment, if we have a nearby catchment, because in WB, what we are trying to do is the catchment population is known, and we are trying to predict the number of cases with the viral load, because we know how much quantity of raw sewage is coming from that given catchment with the given population. So it is very essential in areas, especially where we have a lot of STPs located catering to that catchment. We feel like we are also suggesting that there is an urgent need for this kind of wastewater-based epidemiological approaches, almost all across the globe, and especially in the developing region in particular. So what basically I would sum up is like we have a miniature now lab established. We learned a lot. It was a learning process for, I guess, for a group, because it was for us to get into this area. But we understood a lot of techniques, and STPs, if we combine it with such kind of marker, we might understand the functioning of the STP and also where is the viral load. Because in India, or the map I showed this before, there is this, the riverine region, the sewer outlets are directly there on the river beds. So in those cases, if we want to know the viral load or from where we want to start the work, that could be an important marker. And I also suggest, if there is a budget constraint, sediment method is something we found is pretty interesting when the dissolved solid is more. So with this, the summary ends. I would acknowledge I have to do that, and that's very important, because during this time, the waste that we were generating, that was well taken care by our campus life director. So before we went for the started the experiment, biosafety cabinet level 2 cabinet we purchased with this project, and that was also important. The vendors helped us even before we released the money they gave us. Many of us are researchers, we know the constraint of dealing with the projects. And then our campus life, like we were disinfecting under UV, as I told, ultraviolet radiation again is important. So that's why I mentioned in the previous slides. So all the waste that we were generating every day, we kept it under the UV, and then the associate director was taking care through our biomedical waste management system. So that's what also we were maintaining very well. And importantly, none of us, me and my team, entire group of scholars, we tested ourselves after the entire project got over. None of us were tested positive. So that was like another important thing I wanted to mention, that if we really handle it well, we can do that. And a few more people, like these are my scholars and the RA who were working with me. There was also a postdoctoral fellow for a short time, Dr. Sundar, and this was Tarat Chandra, Ronnie Rex, and Nihal Mohammed, who was acting as the RA. They are my scholars. They're working on different organic compounds in this region based on COVID-19 situation. And we had the Metro Water team also working with us during this phase. And my another scholar, senior scholar, Mrs. Moitri Mukhopadhyay, she had been helping as a backside. She was assisting a lot of stuff, so I have to appreciate her contribution as well. So the striking part of this entire work is like, yes, we actually need wastewater surveillance everywhere. As Dr. Nag rightly started saying that it's a cost-effective way, definitely. We cannot go ahead, test the 10 lakh population, but we can go and test 100 samples of Stps at a much cheaper cost and understand how to prepare ourselves. So policymakers possibly can take good decisions. With this, I thank everyone for listening to me. I hope there was no internet disruption in between, and thank you. Thank you once again.
Thank you, ma'am. I completely agree with you that this pandemic time, all of the scientific community and researchers have come together with the same question that what can I do? Even I had the same questions when I started working with WPE. And of course, it's a very big learning process for all of us. And congratulations to your team that nobody came out positive. And I completely agree that if we are following the protocols, we will be safe and we will be away from this dreadful disease. So without much ado, now I would like to request Dr. Jiang, who is currently developing an online machine learning model for the application of a data-driven approach to the wastewater-based epidemiology data set for calculation of COVID-19 prevalence in the community. And he has joined us from Wollongong University, Australia, to share his experiences on WBE for COVID-19 monitoring, uncertainties, improvements, and applications. Dr. Jiang, please take the.
Thank you. Can everyone hear me and see my screen?
Yes, sir.
Okay. Thank you, Dr. Nag, for the introduction, and good morning, everyone. So I'm speaking from Australia. Today, I'm going to talk about wastewater-based epidemiology for COVID-19 monitoring, focusing on the uncertainties, the improvements, and the applications. So mostly here I will talk about research from my group, how to improve the WBE for COVID-19 monitoring. I want to start with a bit background about WBE. Although we all know WBE is about the analysis of the measure of wastewater to identify the presence of certain markers. This could be chemicals or could be biological biomarkers. And then we can use that to monitoring the public health. And this normally relates to the average use of certain chemicals or the exposure to certain chemicals. For the infectious disease, that means the prevalence rate. So my research previously on WBE before the pandemic was mainly on chemical biomarkers, because at that time, I think the infectious disease was not a major focus yet. So I was working on illicit drugs, like the use of cocaine, heroin, cannabis, on those things, including many of my recent PhD students have been working on the WBE for the monitoring of illicit drug use. Also, we work on use of PPCP. Those are the drugs that is legal. So like the antibiotics and also like paracetamol. Those are commonly used pharmaceuticals and how their metabolites will show up in the wastewater. And then we can determine the use of those chemicals. We also look at the lifestyle biomarkers or the disease biomarkers. So like how people are eating different kind of foods, or how much alcohol they use, and how much they smoke. So all those can be monitored by wastewater analysis. And those have been my previous focus on WBE. And more recently, I'm working on one project before the pandemic is for the pathogen monitoring in wastewater. So we've been looking at the bacteria and virus, but those are the general infectious agents, not the SARS-CoV-2 yet. And then, of course, in the end of 2019, when the pandemic outbreak starts, we move on to SARS-CoV-2. And I think probably from that point on, the scientific community and the general public suddenly realize there is such a research field called WBE, and many universities have been joined in this research efforts. I just checked there is one dashboard for the WBE for COVID. Currently, there are 263 universities doing research on WBE for COVID, 55 countries, and including nearly 2,700 different sampling sites. So this is really a booming time for the use of WBE, and we see lots of excellent work being done in this research field. Many excellent papers published on this. So I will share today many related to the early warning of COVID-19 here, right, for using WBE, because WBE in comparison to the clinical testing, the conventional approach, it has some special features. The first one is about the population. So clinical testing is individual testing, but WB is for population. So that means we use one sample to represent the zero catchments. And these zero catchments could be quite large, could be one whole city, or could be many suburbs being covered in that wastewater sample. Of course, there are sub-catchment level, like people working on take the wastewater sample for the campus, like a university campus. There are some good studies on that. And also people even use this just for the buildings, like for the dormitory on the university campus. So there are different levels of application for the population monitoring of COVID infection. Also, WBE in comparison to clinical testing, it's a low-cost solution. As you can imagine, we are processing a very small number of samples. So that means the time, the resources, and everything needed will be much, much lower. Also, one important feature of WBE is actually the early warning. So that means the virus will show up in the wastewater before clinical testing can capture this. So this is because the viral shedding actually starts before the symptoms onsets. That means when the people being infected, they found some symptoms, and then they will go for the clinical testing. And then they may find out they are positively infected. But for the wastewater, they won't need the identification of the symptom. Because once the person is infected, they may start to shed the virus in the wastewater. And then if you keep on monitoring the wastewater, that means you will be able to very soon catch basically the onset of the infection in the population. In general, this could be at least 7 days before the clinical testing results. So it's a very useful tool for the early monitoring. But here today, I will talk about what we have done for this. The first thing is when we start with the working on using WP for COVID-19, we look at what we can do to improve this. Although the theory of WBE it's easy to understand, but there are lots of uncertainties, or there are different factors we need to consider to achieve the best results from this. So if we look at this flow chart for WBE, we can see from the virus being excreted by the patients to the transportation in the sewer system, in the wastewater collection system, till to the sampling, how you take the wastewater sample, and then you will do the analysis. So what kind of analytical methods will be used, and then you will do the estimation. So there are different steps involved in the WBE, and each of them will have its own uncertainties. So here I just summarize the different WBE processes in this table, and also what are the different factors will be affected the accuracy in this table, and how we estimate the uncertainty. And then by considering all this uncertainty together, we can basically estimate the propagation of uncertainty, like from the shedding of the virus or from the decay of the virus, etc., to the overall prevalence rate. So I won't go into the details, but I will just jump to these conclusions and also what we have found from here, because you can just read the paper for how we develop those equations. So first we look at the virus shedding. This will be related to how many people of those infected will be shedding virus, because not everyone infected will be shedding virus to the wastewater. And also for each person, their shedding magnitude will be different. So we collect lots of data, I think 1,000 data points about the shedding probability and magnitude. And then we obviously can decide on the uncertainty associated with the excretion, so which is calculated for the first step. And then after the excretion, virus enters into the sewer system. So during the in-sewer transportation, obviously the virus will go through decay, or they will be removed off from the wastewater. So during that process, we need to look at the decay kinetics. But here, although we say virus decay, but actually it's the RNA decay, because we use RT-qPCR mostly to detect the virus. So we focus on the RNA decay kinetics. And also, of course, the wastewater properties, the sewer conditions, those will affect the decay. And according to the first order decay kinetics, we can estimate the decay uncertainty here. And then we take samples from either the wastewater treatment plants at the influent points. Sometimes we take samples from like manholes or sewer pumping stations. But there are different sampling strategies. So there could be just a grab sample. So you just go to the sampling site and drop in the sampling bottle and get one sample. That's called grab sample. There is another more useful sampling strategy is like a time proportional sample or flow proportional sample. So that means you take samples at different time points and then mix them together. That will reprint like over 24 hours. Or for flow proportional sample, you would use the auto sampler, which also measures the flow rate of the wastewater. That means when the flow is higher, you take more sample. When the flow is lower, you take less sample. So that will be corrected for the flow. So the different sampling strategies have different impact on the final estimation accuracy as well. And then when you got the wastewater sample, we come back to the lab, we will do the RT-qPCR. We use maybe filtration, centrifugation, or different methods to do this analysis. And here you will basically, we considered the different concentration methods, different extraction methods to extract the RNA, and also consider the different detection assays, like different primer probes, etc. So all those will include an analytical uncertainty as well. And then the final step is the estimation. So you can use the total flow of the sewage or using the daily water use. That two different approaches we consider here. So by using this approach, we decided that the overall uncertainty for the WBE is around 20 to 40%. So it's reasonable. Like I said, WBE is for the average prevalence rate. So we will talk about for the whole population monitoring. And this uncertainty is 20 to 40%. For the excretion, what we found, if the infected patients is more than 10, then the excretion uncertainties becomes unlimited. Because if this number is quite low, then you may have a quite high uncertainty for the excretion. Like I said, not everyone is shedding virus, although they are infected. Also, the shedding magnitude is different. Also, we found in the analytical stage, if a surrogate virus can be used for the internal or external standards, then that can greatly minimize the analytical uncertainty, which contributes greatly to the overall uncertainty. So that's our first step. When we analyze the use of WBE for COVID, we identify those uncertainties. Then we start to work on the different uncertainties, try to reduce the uncertainty, improve our understanding, and so the WBE can be more accurate. So the first work we did is we look at the different shedding sources of SARS-CoV-2 in the wastewater. So this has been done through a very systematic literature review to check what could be a potential source of SARS-CoV-2 in wastewater. So we look at the feces, which is a obvious, which is also considered as the major source of SARS-CoV-2 in the wastewater. So what is the concentration in feces? And also what is the shedding probability? So like in feces around 4.5 log per gram of feces, and the probability is 54.5%. So that means half of the patients tend to shedding virus in the wastewater, and they at that magnitude. So there are different literature. Roughly, the data is comparable. For urine, there is not much. So that means urine can be negligible in the considering the source of the for the shedding. For sputum, this is quite variable, but the concentration, as you can see, is much higher than the feces. So this can be up to 11 log considered to the 4.5 log in the feces. In another study, it's about 7 to 8 log. It's still quite high in comparison to the feces. Also in saliva and blood, blood is quite low. Saliva is also not that high. So through this analysis, we found that actually with the potential sources, so the feces and sputum, those two could be the major sources. But current study, mostly they only consider feces as the shedding source. So there might be a problem there. So that's why we did quite some analysis of all the data from different countries, from different labs. And what we found is the detected concentration in the wastewater is much higher than the theoretical concentration when only feces is considered. So that means there must be other like shedding sources of SARS-CoV-2 in the wastewater. So here is the results. We did collect data from Australia, Canada, France, Germany, Japan, Netherlands, Spain, USA. So quite some major studies. And we didn't include all the WBE study is because there are probably around 600 some papers reported on the WBE, but not all of them report the prevalence of in their catchments. So that means their data, they may report just the positive negative of the wastewater sample, or they report the concentration of SARS-CoV-2 but low prevalence rate. So it makes it difficult to be included in this evaluation. So we eventually choose the studies from those different countries. So by analyzing those, like I said, what we found is if we only consider feces as a shading source, then only like around 35% of the WBE data is within the maximum theoretical concentration. So that means there is definitely another shading source. So we then did another simulation by including sputum in the as a shading source. And what we found that is nearly all the wastewater data will become reasonable. So it will be below the maximum theoretical concentration. So that means sputum could be a significant shading source from this. So we also did a further calculation. So like we said, because the shading from sputum can be varying quite a lot. So we just simulate two scenarios. One is at a lower kind of shading magnitude. One is at a higher shading magnitude. And also we don't know how much of the sputum of those COVID patients will enter in sewer system. So we did just a simulation from 0 to 80%. If the shading magnitude is quite high, then just only 10% of sputum entering the sewer will be accounting for nearly all of the viruses in the wastewater. But if the shading magnitude is more moderate, then this percentage is from 30% to 70%. So all those analyses are showing sputum cannot be neglected as a shading source for SARS-CoV-2 in the wastewater. So we also consider many other factors in this simulation, like because the prevalence rate reported normally they won't be able to cover the asymptomatic patients. Also, they're always uncaptured cases. So according to the report, is around 15.6% to 76.7%. So that means this can change the theoretical cRNA. Also, the analytical recovery efficiency. Not all the studies have corrected their concentration using the recovery efficiency. Also, there is decay in the sewer system. So that means what you detected actually would be lower than the actual shading concentration. Also, the shading duration, because the patients will be shading virus much longer than the symptom period. So all those considered together, the different levels of increase or decrease of the theoretical concentration, but overall the conclusion is still whole. Sputum have a very important contribution to the SARS-CoV-2 in wastewater. So that's a first study about the shading source. So we also found that the decay in the sewer system it's very important for the WBE. Although there are studies about the decay of this virus in water or in wastewater. Previously, like our first presenter talked about that study about SARS-CoV, another virus in wastewater as well, or some similar virus as well. But the sewer system is quite different from just the water environment or just the pure wastewater. So we think the impacts, in addition to wastewater properties, the sewer conditions, and also the different chemicals being used in the sewer system, those could impact the decay of virus in sewer system. So for this study, as you know, SARS-CoV-2 is very contagious, so it's very hard to conduct experiments. So we choose to use two surrogate virus, which are also coronavirus, but it's not as damaging as SARS-CoV-2. So we use 12229E, which is a human coronavirus. Another one is FIPV, which is a cat coronavirus. And then we did study in different sewer reactor systems. So we have a gravity sewer system, which simulates the gravity flow in sewer system. We also have a resin sewer system simulating the pressured sewer system. So what we found is there is always a very fast initial decrease of the virus concentration within 1 hour in the sewer system. And then you have a first order decay following the initial fast decrease. So that shows the first hour decay is or decrease is mostly probably attributed to the adsorption. So that is probably adsorption to the suspended solids in the wastewater or the biofilms in the sewer system, which is quite abundant in the sewer system. We also found a clear difference between the gravity sewer system, which has a certain level of DO, dissolved oxygen, and the resin system. So that shows the dissolved oxygen could also have a significant impact on the survival or the decay of SARS-CoV-2 in the sewer system as well. So this study is still ongoing. We are currently wrapping up all the data, and very soon we will submit the paper. So we will give more quantitative information about the survival of SARS-CoV-2 in the sewer system. Then this data can be used to improve the estimation of prevalence rate from WPE. Our third study on the WPE is on improving the analytical performance of the WPE for COVID-19. So for this study, basically we are following this procedure. First, we choose different co-PCR primer probe sets. So include the one from USA CDC, which is the N1, which is the most popular co-PCR primer probe sets. We also used the China CDC N, which is also widely used, mostly for clinical testing, but haven't been tested on wastewater. We also use E subbuccal and N subbuccal as well. So those are the mainstream co-PCR primer probe sets. And then we want to evaluate the sensitivity, the specificity, and the reliability for the quantification of SARS-CoV-2 in the wastewater. So here, we are not just targeting on the positive or negative detection, it's more focused on whether they have a good quantification potential. So what we did is we use pure plasmids for the E gene and N gene of SARS-CoV-2. We also used the SPECT-MOC sample, so we spec those plasmids to the wastewater, and also we tested them on the real wastewater that is collected from a cruise ship and also from different wastewater treatment plants. And based on those, we evaluated their performance. We also did some sequencing, use line-of-pole sequencing on the primer products to confirm their performance or to identify why some primer probe sets is performing better than others. So that gives us some insights on the choice of the primer probe sets. So we'll just jump to the results. So from what we found out is the CDC N1, it's very sensitive. So that means it can identify very low concentration of SARS-CoV-2 in the wastewater. But however, the like kind of a calibration at the low level is not good. In comparison, the CCDCN has a good linear calibration for different concentration ranges. So we found CCDCN is probably the most reliable quantification primer probe sets for SARS-CoV-2 in wastewater. And then we analyzed the we did some sequencing of the primer probe products, and then we basically evaluated two parameters. One is the average nucleotide identification, and another one is alignment percentage at 90% cutoff. So through analyzing those, we got some insights about why the CCDC N is performing better than CDC N1 at lower concentration, and why CDC N1 is quite sensitive at the lower concentration as well. But also here we also in this study we evaluate the different primer probe sets, how they're performing for the mutants of the SARS-CoV-2 virus, because we know SARS-CoV-2 is keep on evolving. So we align the primer probe sets, the sequence, to the report the SARS-CoV-2 genome from the NCBI database, and then we found they have different disalignment ratio and also the occurrence percentage. So this will be published very soon, but I won't be able to go into all the details for this. So that's our kind of research to improve the WPE. And we also did some application kind of research. So the first one is in collaboration with Ahmed Warish from CSIRO in Australia, and also my previous University of Queensland. So the study is about the wastewater samples from airplanes. As you know, that the toileted wastewater will be collected when the airplanes landed at the airports. Also, we take samples from a cruise ship, which has been got lots of cases, and also docked in the port near my university. So it's has been docked near Wollongong, so allow us to collect the wastewater. So in total, we analyzed wastewater from 3 aircrafts from Hong Kong, from New Delhi, and also Los Angeles, and also this cruise ship in Port Kampala. So what we found in that we can successfully detect the SARS-CoV-2 in those wastewater from the airplanes or from the cruise ship. So that means the monitoring of SARS-CoV-2 in aircraft or cruise ship wastewater could help the industry probably return to full operation sooner. So give them a screening tool for the management of COVID for international travelers. So that's the first application study. The second application study is about estimate the prevalence from the WBE data. As I showed in our uncertainty analysis, there are lots of unknown parameters in those equations, and those can induce different uncertainties. So based on those findings, we think maybe we should be using a data-driven approach instead of using the conventional WBE equations. So here we then identify different input parameters, including, of course, the most important one is SARS-CoV-2 RNA concentration in the wastewater, and also the different parameters related to the sampling, the analysis, also the wastewater parameters like temperature. We also consider air temperature, including the clinical testing ratio as well. And then we did some analysis for the correlation of the prevalence rate and those different input parameters. Here we identify some parameters that, in addition to the wastewater concentration, the air temperature and wastewater temperature actually are critical parameters, which are not considered in the conventional study. So from there, then we establish a model to analyze the to estimate the prevalence rate. So we found the NM model is pretty good in estimating the prevalence rate. And also it's quite robust if the input parameters is not complete, because there are so many parameters there. So yeah, like said in the introduction about myself, I'm leading this so-called Asian Pacific Network for Wastewater Monitoring of COVID-19. It's called WATMOC. This is the website. So we are trying to collect more data from around the Asian Pacific region. And then we want to improve this model using the Google Cloud AI platform. So if you have any data on the COVID-19, please share your data, and you're welcome to join the network. So I guess that's a brief summary about our different research here. It's looking to the future for the WBE for COVID-19. There are still lots of things for us to work on, especially for the detection methods. There are still like quite some different results being reported from different labs. The quantitative correlation between the prevalence and the wastewater concentration still needs to be established. Also, we need to consider the mutation, the vaccination, and also the new management and policies, how those can be reflected in the WPE. Still many researches needed. So coming to the last slide, I want to acknowledge the support, mostly my two postdocs and one PhD working on this, but also support from Australian government and many industry partners to my various research projects. With that, I would like to conclude my presentation. Thank you for your patience.
Thank you, Dr. Jiang. I agree with your targets, and I hope that your idea of monitoring the wastewater in airplanes and the cruise ships works, and it is adapted soon so that we can get rid of the spread of these new variants soon. But for now, in looking at the time, I would quickly like to invite Dr. Arora for her talk and talk about her experience on predicting the next wave. Case study of early prediction in Jaipur. Ma'am, could you please limit your talk to 10 minutes?
Yeah, sure. Thank you so much, Dr. Aditi, for inviting me and having me here. I'll just share my screen, and without wasting time, I will just start on with my talk. So first of all, I really would like to thank the speakers, Dr. Parumita and Dr. Jiang, for giving us a very brief but comprehensive information in a very limited time. I know they have been doing a lot of research, and how they have to compile it in 15 to 20 minutes is outstanding. So thank you so much. So here I am, Dr. Sudipti Arora, research scientist at Dr. Bilal Institute of Biotechnology, Jaipur, to present a case study of wastewater surveillance in Jaipur. So Dr. Aditi has already introduced me. I am an environmental research scientist, and I'm also founder of a center of excellence called Prakrit. So my specialization includes environmental biotechnology, environmental microbiology, and molecular biology. I'm working on wastewater-based epidemiology research since 1.5 years now. So I know a lot of you must be very curious to know about this very potential question, and I think most of you have logged in also for today's webinar knowing, just curious to know that when is the next wave coming? Is it at all coming or not? Because, you know, there are a lot of news that it's not coming now. Will it come or not? And what about this model? Can this really predict the COVID-19 wave? How authentic this model is? And really, being an unknown, sometimes we really think that can sewage really predict a lot of things? So to answer all these curious questions, yes, definitely I am here to tell you that sewage, wastewater can tell a lot of stories about community health. So as Dr. Chakrabarti also mentioned that not only it is restricted to pathogens like bacteria and virus, but also to caffeine and drug misuse and also antibiotic resistance. This being one of the upcoming, even more severe problem than COVID-19, I would say. And wastewater epidemiology is a research, is a field of study to track the presence of these bacteria, these viruses like SARS-CoV-2 by simply collecting the sample and testing it for a particular contaminant. So the wastewater surveillance cycle includes, a simple cycle includes that if any particular patient gets infected by the virus, by the SARS-CoV-2, he might not show symptoms, he might be asymptomatic, but definitely he will shed the virus into the fecal material, into this feces. And this fecal material will then go to the sewage of the particular locality, of a particular community. And then we can collect that sample, bring it to the laboratory, test it for the presence, and thus by data-driven modeling and other approaches, we can predict the early signs of COVID-19. So on this approach, we started on with this study. The process of WBE includes sample collection, concentrating the sample, pre-processing the sample for RNA extraction, and followed by RNA measurement and some data analytics. This is a simple process flow. Just and this is already being explained by different expert speakers here. A little background about WBE. So this project, this term is not a new term for SARS-CoV-2. It has been established worldwide for other viruses, for other surveillances also. But when COVID-19 happened in early 2020, this study was initiated by Netherlands to track the wastewater for SARS-CoV-2. Followed by this, UK and USA and other countries have now started national level WBE programs. For India, this study, this research, WBE, was already being done in 2003 for poliovirus surveillance. And then there came different research groups like SRM Chennai, IIT Gandhinagar, and we, including us, started this COVID-19 surveillance for India. And then other institutions like ICMR, CCMB also joined us to develop a national level surveillance program for India. Definitely, WBE gives us a head start of at least 7 to 28 days, minimum 27 days and maximum 28 days head start about the COVID-19 prevalence in a community. So this being the very important indicator for early prediction of COVID-19 so that we can be much better prepared and we can allocate our resources in a better manner. This not only gives us the understanding of the temporal distribution, also give us the spatial distribution of the cases, which community is a hotspot, and this gives us the evidence-based decision making. So it's not only the digital model, it gives us the evidence-based data-led modeling where we have the real data in hand and we can model it to know the prevalence of COVID-19 in a particular community. So definitely this helps decision makers, policy makers, and government officials to get themselves better prepared for strategic allocation of resources, for identification of hotspot, the containment zone, and in complementing with the clinical surveillance, we can better manage the COVID-19 pandemic situation. So these are the national level initiatives around the world, and there are a lot of agencies like CDC and other which have been working on this. So here I am going to share how we started on with this case study, how we started off WBE with what objectives for Jaipur city and other cities as well. So with these objectives in mind, we started on with this research on WBE as early as on 4th May 2020. So when the entire country was in lockdown, we thought of starting this being a research scientist, myself and my team, we all were curious to see how this research, how this field of WBE can be explored in our city as well. So with the objective of evaluating the successful implementation of WBE for Jaipur city to early predict COVID-19. Also, there were a lot of questions coming on in our mind that what is the risk factor involved with the people who are managing the STPs, because there were already a lot of reports published that COVID-19 particles are present in wastewater from the STPs. So now there was a lot of risk involved. People were really scared, and we ourselves visited these STPs. We saw these people in real anxious and nervousness, that what is the risk involved to us, to our life. So we thought of again evaluating the STPs and these wastewater treatment systems in removal efficiency of SARS-CoV-2. Also, India was a little new in this field, so there are a lot of there were a lot of challenge in standardizing the protocol. The protocol has to be quick as well as cost effective, so that we can detect and quantify the SARS-CoV-2 in wastewater. And also, the objective was also to detect various variants of concern in wastewater samples. So this study we started on as early as 4th May, and still this study is in process, where we have collected various wastewater samples from different sites of Jaipur city, as well as few samples were also collected from Uttarakhand state, and including Roorkee, Haridwar and Rishikesh. So these samples were being transported from Uttarakhand to Rajasthan and to our institute. These are the different sites, 14 wastewater treatment plants, which were studied during the first wave of the study, and first wave of COVID-19, and during second wave, some of the plants were also extended. So initially there were a lot of challenges when we were doing it, as in how we will collect the sample. There was a lot of fear also with the team. Team was also scared that we are safe or not. When we are going in the pandemic, we were going out to STPs and collecting the samples. So of course, considering all the necessary available precautions and PPEs and everything possible, we collected the sample, transported it under cold chain transportation, coding it and storing it at 4 degrees. So we followed everything within SOPs. Then the challenge was to pre-process the samples. So as Dr. Chakravarti said, that they evaluated 4 different methods of pre-processing. So we also thought of had the huge responsibility of standardizing the protocols from Indian perspective from here to our wastewater samples. So we did different wastewater processing. We basically evaluated 2 methods. So method 1 was where we filtered the sample, and using PEG adsorption and centrifugation, we did this processing, which took about 7 hours. And the second method was a very simpler method, which included only the centrifugation followed by RNA isolation, which took over a time of 2 hours 40 minutes. So we compared these 2 methods for different sets of sample, and interestingly, we found great results that method B and A showed similar analysis, statistically also similar results. So we can adopt any of the method as required. Then the RNA extraction was being performed using the automated King-Flex machine, and RT-PCR using Alplex. This kit is being approved by ICMR, and we use the same kit for our quantitative analysis. For quantitative detection, to see the genome loads, we used Enodect one-step COVID-19 kit, again approved kit, and we used mainly 3 genes for the qualitative analysis, E, RDRP, and N gene. And for quantitative detection, we used 2 genes, N gene and ORF1 gene. So this was the protocol which is already being published now. And this whole protocol was being done in collaboration with the industry, that is Dr. Bilal Clinical Laboratory, who has the approval from ICMR for COVID-19 testing. So at CIRD, Center for Innovation, Research and Development, this is being done, and we performed. So these are the results from the first wave of Jaipur. So initially, when we started our study in May, so there were roughly very few cases, clinical cases that time. So we can see the green color shows that there are a lot of samples that came out to be negative. And then but only one sample, that is on site 5, came out to be positive that day. So this correlated with the increasing clinical cases that was reported from the same region. So we corroborated our result with the clinical cases and find out that what the authenticity of this model. And we also took some of the hospital wastewater samples to check on the presence of SARS-CoV-2. And interestingly, we saw that in effluent, in a hospital wastewater sample, we didn't get the positive result, probably because of the huge amount of hypochlorite treatments that have been done at that time in the hospital. So probably the decay of SARS-CoV-2 was assumed. Then we also did the statistical analysis between different technologies, because we evaluated around 14 different plants, which were all based on different sizes and different treatment efficacy. So these treatment technology includes sequencing batch reactor, moving bed biofilm reactor, and conventional activated sludge process, which is a centralized system. So we compared these different technologies for the removal efficiency of effluent. And I'm also happy to share that during the first wave of COVID-19, when there were clinically also less cases and genome loads was also very less in the effluent samples, so there we could not detect any prevalence of SARS-CoV-2 from the effluent sample. And we published this fact also in our publication, that Jaipur's wastewater treatment plants are working good in terms of removal of SARS-CoV-2. And as we can see from the statistical, MBBR showed a better removal of SARS-CoV-2 in terms of all the 3 gene removal. So now coming to the second wave result, this study was still continuing, and we have presented these results from as early as 15th February. So as we all know that the first wave was not much during the January. It was like lowered, the peak was lowered, and then on 15th February, as you can see from the gray dots, so there were practically none of the samples from wastewater came out to be positive. And as we were progressing towards the second wave, clearly the red dot showed the hotspots coming from the different areas of Jaipur. So as early as on around first week of March, we saw again one of the hotspot, and then we again corroborated this with the increasing clinical cases. And then as we were progressing, you can see the graph that shows, by the gray graph, that shows the increasing positive cases of Jaipur city. So as we were progressing toward the second wave, which the cases were exponentially increasing, our red colored zones showed the hotspots that were coming in the Jaipur. And clearly, by first week of April, on 1st April, we could see 90% of our samples to be red. That means the hotspots were there. So this gave us the early warning of at least 20 days ahead. As you can see, the peak of the Jaipur cases came out to be on 5th May. And we got it approximately 28 days before that second wave is about to come. So this clearly gave us a clear indication of the faith and usefulness of this model for early epidemic prediction. And again, these were correlated with the case overload. So you can see as early as in February, all the hotspots were green. So green means that these all are the negative result, and we could not find any hotspot in these areas. But as we were progressing on 27th February, one of the sites came out to be positive. Continuing with it in April and in May, the other sites started coming positive. So clearly, this corroborated with the high case overload. Again, this graph shows the quantitative loads of the various wastewater samples collected over the timeline from February till June. The green curve shows the increasing positive cases. The black curve shows the quantitative load of N gene, and gray curve shows the ORF gene. So we quantitated 2 genes here for the quantification of the genome load, which clearly again gave us the warning of 20 to 25 days with increasing genome loads over a time period. So again, another interesting observation that we were able to find in the second wave is that now the sewage treatment plants, the wastewater treatment plants, at such high active case loads of 6 log or 5 log, and when the COVID cases were also really high during the second wave, these treatment plants were sufficiently not able to decay the SARS-CoV-2. And you can see here, the first picture shows the N gene and the second picture shows about the ORF gene. So on different sites, the removal efficacy of these STPs were shown here, which showed that site 2 and 3 showed better removal efficacy as compared to site 4 and site 8. So this again may be correlated to the treatment efficacy of the plant. So again, the important findings from this study of Jaipur is, yes, firstly, we are able to successfully apply this model in Jaipur city, especially when I talk about Jaipur city. Why I am highlighting this is the sewerage network of Jaipur is not as developed as you can see in the developed countries. Here we have fragmented systems and we have very less wastewater treatments plants, and very few systems are connected to the main city line. So at such fragmented systems, there was a question that whether WBE would be successful here in Jaipur city and from the Indian perspectives. So yes, we were able to successfully apply this model as it was applied in the developed countries. So very happy to share that we got good. In spite of, we were able to cover only 60 to 70% of the Jaipur. We didn't even cover the 100% of the city. Then also we were able to predict the upcoming wave. That's the beauty of this system. Secondly, the sensitivity of this detection were based upon combination of 2 kits. So here we use 2 different kits, because if we use any one of the kit, there is a probability of false positive or false negative. So to rule out and to make the system even more comprehensive, we use 2 different kits so that a better system can be prepared. Then there were some apprehensions earlier in early 2020 that this SARS-CoV-2 won't be able to survive at a higher ambient temperature. So when there will be 40-45 degree temperature, this virus will decay. But our results, we were the first one to publish this fact, this study in IWA Journal of Water Science and Technology, that in Indian climatic condition at ambient temperature of 45 degree, we were able to detect SARS-CoV-2 from wastewater. That means this was another publishing fact. Then again, during the second wave, when there is a higher active load, wastewater treatments were not able to remove the genetic load in spite of all the tertiary treatment technologies. Even UV and chlorine were available as a disinfection. So this we are again our future studies are underway to further investigate this matter of fact. And again, the NSG, we also did the genomic analysis of our wastewater samples from February till June samples, and we were able to identify the designated variant of concern, Delta variant from wastewater sample in Jaipur city. And soon we are going to publish. The manuscript is already under preparation for all these data, facts and figures. So yes, interestingly, WBE studies have been proven successful from Jaipur and also from Uttarakhand state. And these are some of the publications that we were able to publish and some of the news articles. And again, besides this Jaipur, we are also working on one of the international project which is being funded by GCRF, where we are doing the citywide surveillance for Nagpur city. This project is underway, and sharing the result of this project is beyond the scope of this webinar, but soon we are going to publish all the data from this project as well. And also, I'm happy to share and also would like to acknowledge the efforts of Dr. Jiang for making this WetMock network. And this is the Asia Pacific network for wastewater monitoring using data-driven approach. So we are one of the potential partners here, and I'm highly grateful to Dr. Jiang for this opportunity. And I'm sure that our research will really help the society for the successful application and would contribute towards the favorable outcome of the society, especially in this pandemic. Thank you so much. Here, I would like to acknowledge the internal neural grants of Dr. Bilal Institute of Biotechnology for funding this project. This was funded by the IRSC of the Institute, and also our team, enthusiastic young researchers team who are willing to work non-stop and during this pandemic also. So highly appreciate their work, and thank you so much for your kind attention. If you have any queries, I'm happy to answer. Thank you so much.
Thank you, ma'am. Queries maybe we can take at the end of after every talk is over. But I must add to this wonderful talk that this experience of our team was really exhilarating for all of us. And when we saw this data unfold before us and soon. I know that this results would be out, as you mentioned. I'm sure everyone can appreciate how much fun we had while this study was being done. So quickly, for discussing the far-reaching implications of the early prediction, I would like to request Professor A.B. Gupta sir, who has been a mentor for us and a collaborator in our endeavors to monitor SARS-CoV-2 in Jaipur city's wastewater. Please deliver your talk, sir.
Are you able to see the PPT?
Yes, sir.
Oh, thank you. I won't really take much time because we are working in a same group. You have already seen all the detailed results from Dr. Sudipti. And so many of the things I would simply escape. I'll just try to concentrate on some of the things which are still probably left out. We always talk about the urban areas when we talk about sewage treatment, and then because that's an organized form of sewage being collected and treated. But in Rajasthan or in general in India, there are lots of areas, especially the rural areas, which are connected with septic tanks and some with fecal sludge plants also. So with the septic tank being underground, the things are very different. But once that sludge is being brought up and treated in such open sludge drying beds, then the issues can be for the rural areas as well. So we have to take care of rural areas. Another very common issue in India is reuse of sewage for irrigation, which is very, very common. Like in my institute itself, where we have one RBC and two MBBR units, and we covered these MBBR units during our study, and the results have been shown by Dr. Sudipti already. But we made a group of around 30 researchers from across the country during the very early process of cases being reported. The initiative came from IIT Delhi, and there were people from at least 20 different institutions from across the country to bring out certain issues and take some immediate measures. That included a lot of standards being revised or recommended for revision, but certain immediate measures to be taken, like maybe MBBR is still not sufficient, though. So it is told that results are very good, but we may have to go for membrane bioreactors, especially for the hospital sewage, because we proposed very strict standards for hospital of 10 coliforms per 100 ml, so as to take care of any other associated pathogens. And for otherwise domestic sewage, we had put up this, recommended this unit to be 100 instead of the present 1000 coliforms per 100 norm. So another thing which is neglected is always that the sewage is analyzed, but not the sludge. And sludge is dewatered and then, I mean, we do a lot of things for sludge concentration, and you cannot really escape that. So that also has to be touched upon. And some of our studies on hybrid disinfection have yielded very interesting results, where we found that there were so many opportunistic pathogens coming from nosocomial origin into the main sewer trunk. They were highly resistant to chlorine and highly resistant to UV also. But when we tried this hybrid method of chlorine followed by UV or ozone, which we have been doing for last 7-8 years, we found the effects to be much better. But we have to still evaluate for the fate of coronaviruses in it. But one of the plants that was shown by Dr. Sudipti, we have been studying it for last 7-8 years for many other things apart from just corona monitoring. And then we have found in the past Morganella, Klebsiella, all being even resistant to the cholesterol, which is the last resort antibiotic. And these are the points of high concern. So just a few things. This has already been covered, that how this all began in Holland as well as Hong Kong reports. But Sudipti has already again told you about the studies by us in 19 STPs of Jaipur and Haridwar. In the first wave, we had the presence of virus only in the raw sewage. But in the second stage, second wave, when the load was very, very high, both secondary and tertiary treated samples indicated the presence. And that really reflects the importance of adequate disinfection measures, which are to be studied in details. But at the same time, as Dr. Sudeepti indicated, when she brought the first few samples, many of these samples were showing negative and they were coming directly from the hospital. Then we decided to go for dechlorination at the point of collection itself. And then the virus presence was shown. So we normally talk about preserving the samples after bringing it to the lab, but it has to be treated even before bringing it to the lab. So that's very important. And as Dr. Jiang indicated, the decay in the sewer lines was based on many parameters, including the DO. Well, in places like Jaipur, where the ambient temperatures are high, normally we have, and the sewage is very concentrated, we have almost zero DO throughout. So, but the more important thing could be the presence of residual chlorine, because a lot of hypochlorite is used for even cleaning the streets, and all these wastewaters finally reach the sewer system, and hence it is also very, very important. Another issue that was brought out by, again, Dr. Jiang was very, very interesting, that sputum had its concentration or contribution. And that also puts up a question which was earlier shown by Dr. Banerjee, that, or Dr. Chakrabarti probably, sorry, that we did not see any live viruses in the treated sewage. But with the sputum contributing active virus, it has to be really seen in, especially near the aeration tanks of the sewage treatment plants, where it can go into air and directly inhaled. So that risk also has to be assessed. Another thing is the risk due to sewage irrigation, with under the new context, where we can have many more parameters apart from the routine bacteria of concern for the risk due to, like, maybe irrigation being carried out with it. Then another thing is the change in microflora. We took samples from inlet and outlet, but we did not take samples from the biomass. And that is again very important, especially the fixed film biomass. The same treatment plant we studied, and we did detailed metagenomics, and we found a very interesting thing. There was no Vibrio cholerae in the inlet and no Vibrio cholerae in the treated samples whenever we assessed it in the past. I'm talking about a non-corona scenario, but the biomass predominantly indicated the presence of Vibrio cholerae. And hence, the film forming tendencies of certain surrogates are very important to understand, along with that of simple assessments of inlet and outlet streams. So that is also very, very important. And ultimately, you have to create a network, and it can be taken up under the smart city program, which our country is pursuing, that sewage based epidemiology. It will have a lot of indications, but ultimately this should lead to the new standards. Say, for example, very recently we tested one of our samples few months back, and the BOD was 3.5 as against the normal around 10. The reason was, the moment we started our surveillance and found some raw sewage having corona virus, we started doubling the chlorine dose. And that is a similar scenario. There's similar scenario across the country. And hence, the presence of residual chlorine all across the systems has also to be analyzed. especially in the sewer lines, because chlorine will be more or less consumed in the line itself. And that is one of the reasons as to why probably the attenuation was much higher than the prediction as shown by Dr. Jiang. So this biomass, and then it should result into better norms for hospital, municipal sewage, because we don't have any separate systems still, though half of the country has separate hospital sewage treatment plants, but ultimately the sewage goes into the main trunk. So then the possibilities of wastewater-based epidemiology has to be increased further by putting up more parameters as indicated by all the three speakers in details, maybe some PPCPs, the change in antibiotics during this period with predominance of 2-3 antibiotics, maybe a couple of antiviral being injected, that has also to be indicated along with the surrogate chemicals or microbiological science, and so as to carry out another scientific based risk assessment. And we have been carrying out nasal swab results versus sewage virus results, which probably will give some more clues. But virulence is yet to be fully established for viral presence in treated sewage as well as air near the STPs. So finally, a lot of collaboration should be made between morbidity and mortality data based on the swab and the reported results, and then we have to try our results of sewage as well as sludges to find out any real collaboration between the two. So that is all from my side. Thank you so much.
Thank you, sir. That was really, there were some really good questions over there. I think we can open the floor for questions now. If there are any questions from the audience? So I have a few questions. Maybe we can start with those. So the first question is for Professor Chakrabarty. Ma'am, you mentioned that you have collected samples from the pump stations as well as from wastewater treatment plants. So I would like to know, did you find any differences between the loads, viral genetic loads in these two sites, these two types of samples?
Can you repeat what are the two types you said? Sorry, I was wastewater treatment plants and pumping stations, you mean?
Yes.
Viral load was definitely more in the treatment plant samples, but in some cases it was some of the pumping stations, especially from northern Chennai, they were having almost similar load.
Okay, ma'am. And ma'am, there was one interesting point in your talk that you have observed increased loads after lockdown in case of riverbeds. So you saw that open defecation was increasing after the lockdown was taken over, like in post lockdown phases. So I'm very interested to know, why would that be a difference? Like, what changes in the amount of open defecation, or why is this happening?
See, what I suspect is, like, most of the time lockdown phase, the slum areas, maybe those could be a floating population. So during the lockdown phase, they were not there and they came back. That could be one reason. And mostly in those low income group areas, they might be not having a proper sanitation facilities. But actually that is not the case. But then also this practice. And the third possibility is a fear, like, okay, during those phase, maybe people were not going out much. Because we didn't observe much of this open defecation during the lockdown phase when we were collecting, but definitely we observed that post lockdown. Yeah.
Yeah, definitely, because this is a very interesting observation that you made there.
Yeah, especially for the river belts, I think
Right.
it is important. Yeah.
Yeah, because although we are having Swachh Bharat Abhiyan and making many toilets, but I think wherever open defecation is there, lockdown might not affect it. So that was very interesting to see. So my next question is for Dr. Jiang. So I would like to ask you, sir, that why do you think there is different early detection periods? There are different durations. Some of them are reporting 7 days early warning. Some of them, like for our group, we were able to detect it 18 days. So there is a wide range of the early detection period. So why do you think there is a range and not a a more uniform period of early detection?
Thank you for your question. I think for the advantage of early detection, this is all depends on like whether you can detect that signal at the very early stage when there is very low infection case in the catchment area. So whether the method is sensitive enough, and also depending on the shading uncertainties, because you have if you less than 10 cases in the catchment, that means you only got probably half of the people like a shedding virus, and also the magnitude is not very high, then you may not be able to capture that. Although in theory, we should be catching that much early. So that means in the best case, you may be able to catch once there is one case in the community, but in most likely there will be more than one case. That means after the transmission has accumulated to a certain level, you will be able to detect that in wastewater. So there is a different level of like an early warning period. This is also related to the sewer system as well, because even in like in Japan, even in the very highly developed countries, not all the houses are connected to the centralized sewer system. So I remember the number is only average is only 60 or 70% in Japan, but of course in other countries like in Australia, probably 90%. So that means if those infected people, they are not connected to the centralized sewer system, you won't be able to capture their wastewater. So that also creates a another like a barrier for the WBE. So I guess I also was having this one idea is maybe like for countries without like a very broad sewer system, they may be able to use the water channels like rivers or other water bodies as a approach. Instead, it's not wastewater based, but it's still like because those wastewater may find a way entering those water bodies. So by monitoring the river or those lakes or other things, they may still be able to find the correlation there.
Okay, so yeah, so to your point, I would like to share here that even though in India we do not have so much of a connected sewage system and it's more of a fragmented system, we were still able to detect it 18 days before we could actually see a rise in the cases. So that might be one of the points that you're mentioning, and it's a good idea to cover up surface waters as well, where open defecation is a common problem. So that's a very good point that you have risen here. There was one other question, sir. One question is that could you see any correlation between influent genomic loads to the decay caused by biofilms?
So far we haven't done such research about the treatment. We only analyzed a few sample, like from the cruise ship sample, because there is a MBR system on the cruise ship treating the wastewater. what we found in the effluent from that MBR reactor, there is no like a residue of SARS-CoV-2 in that case. I think also we analyzed a few from the local wastewater treatment plant, and in those cases, because in Australia the prevalence is not very high, so the removal efficiency is quite good, so it's adequate. We didn't see any cyclo in the treated effluent here.
Yeah, because would you like to speculate that if the load were high, what would you have expected?
Yeah, we I guess for this the concern is mainly for the active virus. If it shows up in the effluent, it will be a concern of transmission. But currently, most of the detection is based on RNA. That is not based on the culturing method. So like for our sewer study, maybe come back to that effects of biofilm. That study using surrogate viruses based on the culturing or based on infectivity analysis. So it would be quite different from what we talking about for the WBE, because WBE is about like the total number. We are not concerned whether it's active or it's decayed in the wastewater. We are concerned about the total number of the virus. So co-PCR would be the best method. But if we are more focused on the water quality or the biosafety, then we should be looking at the using infectivity test for SARS-CoV-2.
Okay, sir. So this question has just come for for Professor Paramita. Ma'am, the question is that could you see any differences in the inhibitions or inhibitors in your 4 processes that you have done for processing the wastewater samples? Professor Paramita? Maybe she is away. Maybe we can take another question. So there is one question for A.B. Gupta, sir. Sir, you mentioned that the wastewater which was treated outside of hospitals was negative for the presence of viruses. So do you think that if the virus load is increasing with increasing number of patients, there is a risk that a live virus may sneak out in the dumped wastewater?
Definitely, and that is one of the concerns that I also raised, despite the fact that in the first wave, we did not get any corona presence in the treated sewage. You see, in India, even if the hospital is wastewater is receiving some treatment, ultimately it's going into the common sewer. And we collected samples which were getting a lot of samples from hospital. But right outside the hospital, when we took some samples, because a lot of hypochlorite was used for floor cleaning and many other things, that perhaps inhibited the virus then and there. So the moment we started nullifying this chlorine at the site itself, and then we would bring it to the lab, then the positive indication was obtained. So we have to be very careful that pre-processing does not mean the lab pre-processing only. At the time of collection itself, it may need some pre-processing as I indicated.
Okay, sir. Thank you, sir. I think we can take one more question. This question is for Dr. Sudipti. So the question is that there are so many studies which, even from India now, which are stating that WB is being able and it's very useful tool. So why do you think it is not there already being applicable in the cities of India? Do you think there are any limitations, and what limitations they could be?
Yeah, so basically, from COVID-19 perspective, only we came up with this, a confirmatory kind of sure kind of model. Before that, we were not able to trace any such studies related to like it was more for pathogens. We have been doing wastewater for a lot of pathogens, for particularly bacterial pathogens. Lot of studies have been undergoing from last 10 years. But from virus perspective, for the first time, there was just 1 or 2 papers we were able to find out. That study was from Bombay for polio virus. And then from that, there were not enough studies for virus surveillance. And then during COVID-19 only, lot of studies came. Probably the challenge is that when India is concerned, the sewerage network, again, as I said in my talk also, there is a apprehension about the sewerage systems, that how well developed they are, whether we'll be able to trace them back to the prevalence or not. So a lot of apprehensions were there. But yes, the good part is now, since we know that it is a very promising tool, as A.V. Gupta sir said, that we have to come up with even stricter norms now when we are talking about sewage. And definitely better disinfection systems are required, not only for hospital, but for even municipal effluents now. Thank you.
Okay, thank you, ma'am. In the spirit of time, I think we should conclude this wonderful session here. This was really informative for me, and I'm sure for most of our audience who did not know that WB could be so useful, especially in case of country like India, where cost-effective measures are so much required in such a populous country. So with this, I would like to thank all our honorable speakers, Professor Chakrabarti, Dr. Jiang, Dr. Gupta, and Dr. Sudipti Arora for joining us and sharing their experiences with us. I would like to thank all the audiences who have joined us today on behalf of Dr. Bilal Institute of Biotechnology for making this a wonderful webinar. Thank you, everyone. Hope to see you soon.
Thank you.
Thank you, Dr. Aditi, for moderating this session very well. Thank you, everyone, for being a part of this international webinar. I'm really thrilled to see all of the researches that have been carried in different parts of the country as well as world. Thank you so much.
Thank you.
Thank you. Please.